flow are extremely important. Animals are common when large channels exist in groundwater. And these karst aquifers have fabulous diversity. You can have blind crayfishes, unpigmented salamanders in some of the big cave systems, really unique biota. Fishes that moved from the surface water into it, lost their ability to see. Some really interesting habitats there. We've also have some situations, we talked about it there's a sidebar in the nutrient cycling about Mobile Cave, where there's actually a sulfide-rich spring that comes into this cave way underground, but there's also oxygen in the air of the cave. It's connected enough to the surface that oxygen can move into it. You have sulfur oxidizing bacteria. They're basically sulfur oxidizing bacteria, chemoautotrophs, so they're fixing carbon dioxide, and they're a rich carbon source for the macroinvertebrates, and there's a whole endemic suite of invertebrates that depend upon that. And they went ahead and used isotopic analysis to trace whether the carbon from the system's coming in, washing in from outside, or if the carbon's being fixed right there. And so, like we talked about, tracer natural abundance of isotopes. And they figured out that it is a food web that's based just on this one little cave. And so there's some really interesting things that are going on in the groundwater. Unfortunately, it's the most difficult habitat to research, so there's probably a lot going on that we don't know about. But that means there's a lot for you guys to do as scientists in the future.
How do organisms get groundwaters and established there?
Through surface waters usually. And so we have just in this region, we have at least 3 species, probably more, of either crustaceans or planarians, microcrustaceans or planarians. And this is an ocean, right? And they were derived from marine species that as the oceans went away, they worked their way down into the subsurface and did well there. So, there's a trade-off. There's almost no predators down there, right? So you don't have to grow very fast, but the food is really low oftentimes as well. So there's this trade-off, but organisms have evolved to use most of the habitats on Earth, and that's just one of the examples where they did. It's a good question. So if we look at the relative rates of respiration of ecosystems, this is a logarithmic scale. And it goes over 15 orders of magnitude, right? And we see that, sort of the extreme is this secondary sewage stage of sewage treatment, right, where you just have these extraordinarily high rates of metabolism. And perhaps in soils you can get up near there. Lake sediments and soils are not that different from each other. But relative to those things, subsurface aquifers and the very deepest aquifers, ones that are the furthest away from surface sources of organic carbon, have extremely low rates of metabolism. Basically 10 orders of magnitude lower than the highest rates that are seen anywhere on Earth. And so things are extraordinarily limited by organic carbon down there oftentimes. And there have been studies where they've actually shown that the microbial community is growing and dividing perhaps over the order of hundreds of years, right? So they're growing extremely slowly down there. It is also potentially the only place where there is a ecosystem that's not based on photosynthesis, either directly or indirectly, because there are places where there's hydrogen generation from the rocks, and they can use the hydrogen and combine it with CO2 to create methane, and that gets energy so they can actually fix some of that carbon. And through the methanogenesis then becomes the source of the whole ecosystem carbon in that system. That is in contrast to what we just talked about, the sulfide coming out in the cave. The sulfide comes from the groundwater, but the oxygen ultimately came from photosynthesis, right? So they're still dependent upon photosynthesis indirectly at least. We'll see how common this is, I think, over the next 10 or 20 years as scientists do more and more research on this. It's not totally nailed down yet. Let's say one more thing, though, that even though these deep aquifers have extraordinarily low rates of metabolism and the microbial biomass is low per gram of sediment, because of the extent of these on Earth. That is, if you think about the biosphere, from the active part of the soil up to the top of the trees is what, maybe 10 meters, 20 meters of this little skin that goes around the globe terrestrially. Or you have open oceans that are fairly nutrient poor, then you can have a couple hundred, maybe a couple kilometers of depth. But the deep surface groundwaters are more like 10 kilometers, and they're under every bit of the Earth. And so then we know now that the biggest biomass of biologically active organisms is microbial, because this tips the balance away from trees on the land and phytoplankton in the ocean to bacteria basically throughout the world. Okay, so we've got a number of ecosystem things that are occurring in streams. The idea of flood pulse concept, the lockedness versus the topness, so production inside versus production outside the stream, nutrient spiraling in the river continuum. So the idea that stream ecosystems are somehow harmed by floods is an old and perhaps somewhat outmoded view of the world, because floods are natural, although they are a disturbance. And we had a question on them in the last exam, is how much of a disturbance they are. They also are something that's occurred very commonly. And it's been more recently realized that it's an important part of the system. And there's several reasons for this. And one is that it moves materials in and out of the riparian zone, right? Because a typical river or stream has side channels. It has wetlands perhaps, and particularly in the larger rivers, large wetlands, oxbows, all these things that are mostly disconnected from the system, but at times they become connected to the rest of the system. And so it can be an important movement of materials. And so, for example, in 1993, when we had large floods on the Missouri River and the Mississippi River, a number of the native fish species were able to reproduce more successfully than they had for many, many years, because the flood essentially broke down these traditional small constraints to it moving out on the floodplain, and the fish were able to get out there, reproduce, and get back in and the larvae were able to get back into the main channel. And so the idea that there's these systems that regularly flood, and this can be important habitat for species, is becoming more and more understood. We talked, and actually James talked about that a little bit in his talk as well, right? The idea that there's, in the southwest rivers, there's a lot of non-native species that are invading and taking over from the endemics. And one of the ideas is that if you bring back the natural flow regime, the non-native species are not adapted to it, and you can tip the balance in favor of the native species. So it's being used as a management tool to bring systems back to their natural systems and conserve biodiversity. Another interesting place where this has been taken advantage of by organisms is in the Amazon River. The Amazon River floods every year, and it moves out into the riparian forest along the edges of that large river. A number of species take advantage of that time of year to migrate out and reproduce. The most spectacular behavior related to that, though, are the fishes that use fruits. They're piranhas that use fruits from the trees that are out in the flooded forest. And the trees actually produce fruits that they dip down into the water. They grow, so they grow down into the water. So they're basically have co-evolved with these fishes. The fishes come up and eat the fleshy fruit, ingest it, and then swim off somewhere else and excrete the seeds out and plant them on the bottom, essentially, with a packet full of fertilizer. And that's a very interesting adaptation related to this flood pulse that comes through the system. And this is where the idea of the importance of this annual pulse, or this normal pulse driving the system came from. But it's expanded then to temperate rivers. And it may even be true in small streams, that it may be a really vital part of these systems, because if you walk out and look at a small stream, you'll see these side channels, you'll see these small micro wetlands, if you will, all these different things that are sort of connected, but only when you have high flow, and it's part of the system, and then they'll be maybe connected through groundwater at other times. So it's a change in our view of how stream ecosystems work and what drives it. That is, they're inherently non-equilibrium systems. That's an essential characteristic.
We have a question.
Yes, Bob.
How do humans making levees affect the environment like that?
Yeah, so that's a good point. You're stealing my test question. Because you know the answer, right? It's obviously they disconnect the floodplain, levees disconnect the floodplain from the rest of the river. And so they have a very strong negative effect on species that rely on those wetlands and those oxbows and those side channels to reproduce or as vital habitat. In addition,
does that also decrease the nutrients in the prairie and things like that, where it's used to flooding?
yeah, so any habitat that's used to flooding, it basically changes. So the croplands that are on the outside become less likely inundated. That's a central part of the productivity of Egypt, right, is because the Nile flooded regularly and flooded out on the delta and refertilized those fields every year. And then as the waters went away, people could plant and grow crops without using artificial fertilizers or created fertilizers. They could just use a natural fertile system. It led to a stable civilization there for many, many centuries. Now that's going away because the Aswan High Dam is pulling the sediments out, controlling the floods, so there's not floods, and the Nile Delta is sort of retracting because of that loss of particulate material input. Levees have the other negative effect in that when the water does flood and is between the levees, it's going at a much higher water velocity. If it finally breaks through the levee, oftentimes it's moving much more quickly than it would have if it had just spread out naturally and then come back. So it can be much more sort of catastrophic. And the last problem with levees is that they tend to actually be dams that only so much water can get through it. So when we've had these big floods on the Mississippi, for example, upstream of St. Louis had really bad flooding. St. Louis has huge high levees to protect the city, and those basically serve as a dam and push the flooding back and up behind it, so the water backed up. So there's a lot of negative aspects to them. The positive ones being that the cities don't flood, so there's obviously a trade-off there. Okay. That's it for this class. We'll go to allotments and autochthonous production from that. Have fun studying for your lab test tomorrow and working up your lab notebooks. And see you soon. Dreams and the idea of allotthonous versus autochthonous production. And these are hellacious words to spell, yet they're actually fairly similar. And one is auto means self-feeding, essentially, or carbon from within the system. That's primary production. Allotthonous, other, coming from outside of the system. And so one of the key issues in aquatic systems is that where is the carbon coming from? Because then that gives you an idea of what's controlling the food web. And if it's an autochthonous system in a stream, you have more primary production, right? And what's the group of organisms called that are generally driving the primary production in streams? Losers.
I said producers.
I think I said losers.
No,
gainers.
Could be, yeah.
Producers, primary producers, but which are the primary producers? What's the generic term for those? Periphyton, right? It's the algae that are actually associated with the periphyton, because periphyton is a mixture of a biofilm with bacteria and protozoa and algae all mushed together in a layer. But most of the in streams, most of the system production is based on benthic organisms, those that are attached to the bottom. The plankton doesn't do very well in most streams because it's washed out fairly quickly. And if you have more light reaching the system, there's primary production. And so we see these areas where the streams are fairly open and there's a lot of algae in there. And the idea then has historically been, well, these could be areas where the key to the food web is understanding the primary producers in the system. Alternatively, we have other systems with forested where they have large amounts of leaf input, or at least quite shaded much of the time. So in the fall, then a bunch of leaf material falls in, and there's a whole part of the food web that can be based on that. That would be the microbes that break down that detritus, that leaf material, that coarse particulate organic material, and the invertebrates that eat that material. And so that could be the crayfish or the tepulids, called shredders often. Their functional feeding group is shredders. And then that might drive most of the food web. We've already talked about how this might vary seasonally, because we've talked about seasonal succession in streams. And so then you can imagine that the relative importance of a locthiness and a toxinous measurement production is sort of bouncing around. So this time of year, if there's any water in the streams around here, which there's not, this time of year when the leaves are all off the trees, the light's low, so there may not be a lot of production, but there's some production going on. And as the leaves get eaten up over the winter time by the invertebrates that are overwintering in those streams, then the primary production in the spring can come up once the temperatures and light comes up, and the system could become more autochthonous than loctinous. So initially, when people went out and made these measurements, they just took rocks off the bottom and they put them in chambers and they measured the metabolism of those, and they felt that whole stream ecosystems could certainly be autochthonous and driven very heavily by primary producers. And then these methods came out where we actually measured the oxygen in the stream and how it varied over a day, and used that information with the exchange rates of oxygen with the atmosphere, the aeration, the attempt of the stream water to basically become in equilibrium with the atmosphere, right? And so if you know what that flux rate is and how much oxygen is accumulating and being lost in the water column over a 24-hour period, you can make an estimate of whole system metabolism. And that would include things that were down deeper other than the stuff that you got in your chamber. And when those measurements were made, it turned out that most systems are net heterotrophic. That is, most systems, if you remember, we have gross GPP, right? And when we measured photosynthesis, we had gross primary production, we had net primary production, and we had respiration. But you can think of this as in a whole ecosystem, you'd have community respiration, net ecosystem production, and net ecosystem gross primary production. And if you calculate that, the net ecosystem production tends to be a system that is giving off CO2 and using organic carbon. And to have a system over long periods of time that is net heterotrophic, right, means that where does the material have to come from? More of the carbon have to come from. As if you're always respiring away carbon, right, on balance, what has to happen? Where does it have to come from?
A leaf litter.
Right, it has to be coming from outside. It has to be coming from olefin sources. And it could be leaf litter, or it could be particulate material washing in from upstream, or it could be organic carbon that's dissolved in the water that's washing into the stream, that the microbes are using the dissolved organic carbon up as well. And so it could be any of those things. And we'll come back and touch on this a little bit more, because it turns out that in streams it makes perfect sense that at least some streams at some times are net heterotrophic. In lakes, many people have thought that they were autotrophic, but it turns out that more of them are net heterotrophic than it's been thought in the past. And that's something over the last 10 years or so that's really kind of come around to and change in our understanding of the basic function of the ecosystems. The next concept I want to talk about is the way nutrients cycle in streams. And so if you remember back a couple of slides back, I talked about diagramming this little cold water pond, and we have this influx, and then everything's kind of happening in here, and then some of it goes out. In contrast, Jack Webster realized that nutrients in streams actually tend to have a spiraling to them. Rather than being a cycle, like we have, remember we do our nutrient cycles and they have these circles in them. It looked like a wheel. It's actually, as it flows downstream, and the idea is it's a spiral. And he came up with a conceptual idea. Dennis Newbold, a few years later, tacked the term nutrient spiraling onto it, and it gives it a better ring to it. But essentially what you have is the rate that any nutrient can move, this can be carbon, nitrogen, phosphorus, will move downstream. And there's a couple of components to that. One is when it's dissolved in the water, so you can think of something like CO2 or ammonium or nitrate or phosphate that's dissolved in the water. And then it's taken up into particulate fraction. So S sub W is the water part, and S sub P is the particulate part. And maybe the particles can be moved somewhat downstream, or they can be stuck in the bottom. And you can see that generally, you'd expect this stuff that's dissolved in the water to be more mobile than the particulate material, because particulate material sinks out. And this will include actually organisms that are trying to hold on and not go anywhere, right? They're part of that component of it. So if you have an ammonia molecule floating down the stream, and alga takes it up, boom, it stops with that alga. Maybe a mayfly eats it, boom, it stops with that. And then maybe even a fish eats that, and it doesn't move very far, so it's there, and then the fish excretes it, it's up again, and off it goes, right? So you have those two components. And this is important because then it tells us how much nutrient is retained in the stream, how efficient it is at holding nutrients back in the stream. And as we put this in context of global biogeochemistry, how do nutrients move from land through the river network out into the ocean? This component turns out to be quite important, because once the nutrient gets into the water, it doesn't necessarily all make it down out to the ocean. And so you can imagine that if these spiraling lengths are very short, then the retention is very high. And if the nitrogen, for example, is held in the system, the chance that it gets denitrified and blown off as nitrogen gas becomes much greater, right? Whereas if you have long spiraling lengths, then it goes through the system without interacting with the system very much. It's not retained and held back. And even phosphorus can be taken up and deposited, say, in river sediments, as opposed to being shot out downstream. And so these are important issues. We can think then about how maybe the uptake length would vary. So why do you think it is that I've kind of got this here, and then this is short here? Let's say it just flows coming this way, because it's going downhill. What's your guess as to why that I drew this that way?
In the second, it's retained better in the system because it doesn't move as far.
What do you think the difference is between these two habitats? What do you think this? Yeah, this is a pool, right? And what's the difference between a pool and a riffle? Yeah, less water, less discharge, right? So if water moves more slowly, right, the uptake length, the spiraling length is going to be shorter. So there's a velocity component to spiraling. If you have more active biomass on the bottom of the stream that's taking up nutrients, you're going to have shorter spiraling length. It turns out that also if you have higher concentrations of nutrients in the water, because the biota can satisfy its demand more easily, it'll tend to move downstream further, faster. So this leads to this idea of what we're doing when we put in nutrients through additions, sort of say cropland or sewage outfall. We tend to saturate up the system and cause those uptake lengths to get much, much longer. And then you can think of this in terms of people that have done carbon, so they've used stable isotopes, for example, in dissolved organic carbon, and looked at how fast it moves into particulate material, how fast that particulate material moves downstream, and calculate both those things. And then think about things like temperature. How would you think temperature affects spiraling rates? So colder temperature would make longer or shorter spiraling lengths. Longer, why? Yeah, 50% chance of getting it right. I thought you might have a reason for that.
Less biotic activity.
Right, the less biotic activity, so they're not going to take up the nutrients as fast. It's going to get further down. Let's say you have a system that has heavily lighted portions on it. Do you think that's going to have a longer or shorter uptake length?
Sure.
Why?
More photosynthesis.
Right, more photosynthesis, more demand for the nutrients to pull it in, so it's going to shorten it up. So you can see, you can play these games as to where it's going to go. This kind of thing is one of the major areas of research that I've been involved with over the last 15 years or so, using isotopes of stable isotopes of nitrogen to trace the uptake length and processing of nitrogen as it goes to the system. In the first set of experiments we did, we made measurements of uptake of ammonium in 8 different streams across the continent, and looked at how far that ammonium went in the system. So you just put in the isotope, then you measure in the water that decline of the isotope in the water, and you can calculate how long this portion is. And then we did this particular experiment we did for 6 weeks, and so we could actually, and then we stopped it, and we could actually calculate how long it's retained in this part of it too. So you get actually the full retention of the system, the full uptake length. This information suggested that in relatively nutrient-poor systems, about 60% of the nitrogen is retained over each kilometer of the stream channel. But as we increased the nutrients, we weren't so sure what would happen. So the next round of those experiments were with nitrate release as opposed to ammonium, and they were much shorter term, but we used a whole lot more isotope. And to label up stuff, you need to actually have, to use this stable isotope, you need to have a lot if there's a lot of nitrogen around. So we did streams that were in urban, agricultural, and native systems, and we did them at 7 different areas around the contiguous United States, and looked at how far that nitrate went and how much of it ended up being denitrified. So we could sample how much of the isotope ended up in what gases if we're looking at denitrification.
Nitrogen gas.
Nitrogen gas, right, and what's another one that's on the way to it? Yeah, N2O, right? Nitrous oxide, right? Laughing gas, but it's also greenhouse gas. And we looked at those, and we found that the pristine systems were fairly retentive of the nitrogen, but that systems tend to get saturated up as you loaded them with more and more nitrogen. One of the experiments was done up here, just where that little stream goes through Marlatt and Denison, where Denison hits Marlatt up there. There's a little stream that goes by the pig farm up there. And we ended up dumping about $20,000 worth of isotope down the stream in a 24-hour period. So I didn't sleep very well that night, but the experiment worked, and that was the highest, the concentrations were about 15 micrograms per liter nitrate in that system, so they're quite high. It's because they dump their pig waste in the lagoon up there, and then it comes through the groundwater and gets down in that stream. Plus there's a bunch of cropland up, so it's got a real high nitrogen. One of the other things that we found out from this is that although denitrification doesn't make up as big of a portion as much of a proportion of the nitrogen loss that you'd hope it would, it is important because, one, it retains nitrogen, two, some of it doesn't make it all the way to N2. It goes off as N2O, nitrous oxide. And we figured out that about twice as much nitrous oxide is coming from streams as into the atmosphere as thought previously. And so that this change in spiraling length then related to nitrogen loading also results in increased greenhouse gas emissions to the atmosphere. And it makes a difference in the way that the IPCC calculates the global greenhouse gas budget, that there's more nitrous oxide coming out of these streams that are hot spots. So not only is there a problem with adding nitrogen to systems as a pollutant from croplands or from sewage, because it goes downstream and stimulates primary production in the Gulf of Mexico, which then leads to the dead zone, right, and those environmental problems there, because it changes the biotic integrity in the actual stream, but also it's being converted to greenhouse gases as well. So the group that started this, Jack Webster, was also actually involved in all these projects all the way through. Still continues to be something that people measure regularly. The project that Danelle's involved with, Scale Consumers and Low-Tech Ecosystem Rates. We're interested in the rates that are going on in the streams, and one of those things we're interested in is what's the spiraling length, and how do the animals that are in the stream influence that spiraling length. And so we're doing removals of consumers over 100-meter reaches or so of stream, and looking at the nutrient spiraling rate, the nutrient uptake length in each of those reaches with and without the consumers. And then seeing how that whole effect scales up to the entire watershed.